Hello students, I am your educator Dr. Heena and today we are going to study chapter number 8 of 12th standard, that is Human Health and Diseases. This chapter has been divided into 4 parts, and out of these 4 parts, first part we have already studied. Today we are going to deal with part 2. Part 2 of this chapter has the topic immunity. What is immunity? Immunity is the ability of the host to fight and protect against disease causing organisms conferred by the immune system. This is the capability of an individual or an organism to fight against diseases. Suppose if you get any disease, we have already discussed that diseases are caused due to pathogens, that is by microorganisms. When these microorganisms enter our body has the capability to fight against these organisms, fight against these pathogens. This capability is known as immunity, and this immunity is conferred by a well developed system inside our body. This system is called as immune system. Immunity is of two types: innate immunity and acquired immunity. Innate immunity is the immunity that we already have since our birth. It's already present in our body. It is a non-specific immunity. That means it is not specific for a certain type of disease or a certain type of microorganism. This type of immunity is called as innate immunity. Second is acquired immunity. Acquired immunity is the immunity that we get after our birth, that is during our lifespan. And this immunity is specific immunity. That means it is fixed for certain type of organisms. Let's see these immunity, both type of immunity, one by one. Let's read the first line. Innate immunity is a non-specific type of immunity. It is an immunity that is present in an individual at the time of birth, and it works by preventing the entry of pathogens through various barriers. As I said, it is non-specific, so it is not any specific for a certain type of organism. It prevents the entry of all the pathogens. So how does it work? It works by forming barriers. There are various type of barriers by which the body will prevent the entry of foreign organisms into our body. Here in innate immunity, 4 different types of barriers are there. Just have a look at this figure. You can see epithelial barriers. Epithelial barriers mean the barriers which are present in our skin, in the outer epidermis, or in the inner lining of our body. They form the epithelial barriers. Then here you can see some cells, macrophages and neutrophils. So they form the cellular barriers. The soluble molecules are shown over here in the image. They form the physiological barriers. And here some receptors are shown. They form the cytokinin barriers. So different types of barriers are present inside our body, which are responsible for giving us immunity that is non-specific. Let's study these type of barriers in detail. Here you can see 4 different types of barriers. I want you all to note all these 4 types because they are asked in board exams frequently. First type of barrier, physical barriers. As I said, skin, that is the outer epidermis of our body, skin. This is the major barrier. We all know that skin protects us from various types of microorganisms. Wherever there is an opening in the skin, that area is prone to get the disease easily. So first barrier and the most important barrier is skin. Then in physical barriers, second types are the mucous coating of epithelium, which are inside the respiratory, gastrointestinal and urinogenital tract. That is the inner epidermis of these 3 tracts. These 3 tracts are very, very important, and they are in contact with the external environment. So microorganisms can easily enter from these 3 tracts. That's why the physical barriers are present here. Second type of barriers are physiological barriers. Physiological barriers are present in the form of acid, that is HCl, in the stomach, in the form of saliva in our mouth, and in the form of tears, which are released from the eyes. Third type of barrier are cellular barriers. Cellular barriers are the cells that are specially made up of that are specially made for providing immunity to our body. And these cells are the leukocytes, that is WBC. They can phagocytose and destroy the microorganisms. What is this phagocytose? Here, phagocytose means they can eat up the microorganisms. They can carry out phagocytosis. Example of these leukocytes are polymorphonuclear leukocytes, which are also called as PMNL neutrophils, monocytes. Then the killer type of leukocytes, which are lymphocytes, which are the most important type of leukocytes for providing immunity. And there are some macrophages in our tissues. Fourth barriers are cytokine barriers. Cytokine barriers are the special proteins that are released from the cells which are infected with a virus. Now, suppose if there is a group of cells in a tissue, one cell is affected by a virus, what it will do? It will release special proteins, it will release signals, and these signals will tell the other cells that they have to protect themselves from the virus, that virus has entered and now they have to protect themselves. So here what it does, it actually helps the other cells so that they are not infected. And the proteins which are released by these cells are known as interferons. And these interferons will protect the other non-infected cells from that viral infection. So innate immunity is non-specific immunity, yet it is very important for our body as it provides immunity to our body from all type of microorganisms. Moving further, suppose if innate immunity fails to protect us from certain organisms, we have another type of immunity which is called as acquired immunity. This type of immunity is pathogen specific and it is characterized by memory. Memory means our body remembers which type of pathogen has attacked it earlier in life, and this works according to that memory. When the pathogen is encountered for the first time, a low intensity primary response is produced. And if that same pathogen attacks our body for the second time or for the third time, at that time what will happen? Our body will produce a high intensity response. Why? Because it has the memory of the previous attack. We all know that there are certain diseases which are caused once in a lifetime. Let's take the example of chickenpox. We all know that when we were a teenager or when we were small, young in size, young in age, at that time what happened? Chickenpox had attacked most of us. Not everybody, but most of us. And that was the first and only time that we have faced chickenpox. And after that, we never had that disease. Why? Because when the virus attacked us for the first time, at that time our body took a little bit of time to understand that pathogen, that virus, not only about chickenpox but any other disease. Many other diseases are there, even jaundice is there. So our body tried to fight against it. At that time it took a lot of time, that's why the pathogen had won the war between our body and that pathogen. But what our body did, our body kept those cells which it had produced against that antigen, kept it and saved it, preserved it in the form of memory. Now, if at all there was another attack by the same virus after a few years, our body already had the memory cells, so our body attacked before the virus even spread up in our body. And this response is called as secondary response. And this response is very important because it saves us from second encounter with the disease. This response is also called as an amnestic response. Primary and secondary response are carried out with the help of two special types of lymphocytes, B lymphocytes and T lymphocyte. B lymphocytes has this letter B because they're produced in bone marrow, and T lymphocytes has the letter T because they are produced in thymus gland. The functions of these two are a bit different, but they both are responsible for providing us immunity. B lymphocytes produce some special proteins which are called as antibodies, whereas the role of T lymphocyte is to help B lymphocyte to produce the antibodies. See, this is an antibody. This is the structure of antibody. Here you can see each antibody has two light chains and two heavy chains. Here, this red one is the light chain and this blue one is the heavy chain. You can see here two light chains and two heavy chains. That's why the formula of this becomes H2L2, heavy 2, light 2. There are 5 different types of antibodies in our body which are very, very important. IgA, IgG, IgD, IgE and IgM. What is this IG? IG stands for immunoglobulin. Now these 5 different types of antibodies has different roles in our body. The IgG type of antibody is present in maximum amount, around 70 to 75% of immunoglobulins that are present in our body are of IgG type. IgG is again divided into 4 subclasses: IgG1, IgG2, IgG3 and IgG4. So this is the major type of antibody that is present in our body. Second, IgM. Here, IgM antibodies are the first antibodies that are produced when we have any type of infection. Suppose if we have viral attack, bacterial attack, the first antibody that is present in our body, that is produced in our body, is IgM type. IgA type of antibodies are provided from mother's milk, the breast milk. IgE type of antibodies are produced during allergic reactions. And IgD type of antibodies have unknown function. Still, its function is not known, but it is present in our body. So these 5 types of antibodies are present in our body, which provide us protection against different types of microorganisms. Now, when our body responds with the help of these antibodies, that response is called as humoral response, humoral immune response. It is a kind of immune response that involves production of antibodies, and humor means liquid, right? So body liquid is the blood. Blood is the fluid that is present in our body, and inside the blood these antibodies are formed. That's why this type of response, which is due to the antibodies which are present in the blood. Blood means body humor. That's why this response is called as humoral immune response. Now, apart from these antibodies, there are certain cells which are responsible for providing us immunity. We have talked earlier about this. These cells form another type of immune response which is called as cell-mediated immune response or cell-mediated immunity. In cell-mediated immunity, antibodies are not formed. But here what are formed? Lymphocytes are formed. They involve the activation of phagocytes, antigen-specific cytotoxic T lymphocytes, and the release of various cytokines. We have already studied what are cytokines, right? The proteins that are released from virus-infected cells, interferons. So here, if this type of response takes place, this response is called as cell-mediated immune response. Our body is able to differentiate self and non-self. Self means the antigens or anything that is present inside a body, produced by our body. That is self, and something that is coming from outside the body is non-self. So our body has this capability of differentiating between self and non-self. And this cell-mediated immune response is responsible for graft rejection. What is this graft rejection? You might have heard about organ transplantation. When our body undergoes this organ transplantation, our body tries to reject that organ. It tries to reject that organ or part of that organ, which is called as graft. Why it does that? Because it is able to differentiate that that organ is not our body's organ, and it tries to attack the cells of that organ. So what doctors do in these cases? They give us some immunosuppressors so that our immune system is suppressed and it is not fighting against that organ. Okay, now we'll talk about 2 different types of immunity. These are active immunity and passive immunity. Active immunity is the immunity that is formed inside our body. Passive immunity is the immunity that is produced in some other's body and it is given to us. Active immunity is divided into two types: naturally acquired and artificially acquired. Again, passive immunity is also divided into two types: naturally acquired and artificially acquired. Let's see these one by one. What is this active immunity? As I said, this is formed inside our body. So when a host is exposed to antigens, which may be in the form of living or dead microbes or proteins, antibodies, or other proteins, antibodies are produced in the host body. This is the immunity that we all know. Suppose if there is attack of any organism, our body will produce antibodies. So this is active immunity. Now I said earlier that it is of two types: natural as well as artificial. In natural active immunity, what happens? The virus attacks our body and antibodies are produced. Simple definition. But in artificial, what happens? In artificial, weakened viruses or weakened pathogens are given to our body in the form of vaccines. And at that time, what happens? Our body produces response against it. You all know the process of vaccination that we are going to do in detail, but it is a type of artificial active immunity because the virus is not attacking us naturally. Instead, somebody else is putting that virus inside our body, but a weaker one, so that our body can fight against it. Active immunity is actually effective after a lag period. That means it takes a bit time because as we know that our body needs some time to produce cells, antibodies. So it is effective after a short period of time. In active immunity, memory cells are produced, which can produce stronger response the next time the body encounters the same antigen. We have seen this earlier, memory cells. So our body forms memory cells. So active immunity is responsible to provide us immunization in secondary response. If it is natural, it doesn't provide us, most of the time, it doesn't provide us response in the first attack. Examples are vaccines like BCG, MMR. We have heard about these vaccines. Nowadays, so many vaccines are given. When we talk about passive immunity, as I said, this is not formed in our body. Passive immunity is actually the immunity that is formed in some other organism, and that antibodies, because antibodies are formed in some other organism, that antibodies are given to us. So that is passive immunity. You can see the definition. When ready-made antibodies are directly given to protect the body against foreign agents, it is called as passive immunity. This immunity doesn't need time because already the antibodies are formed. So this type of immunity is immediately effective after antibodies are given. Here memory cells are also not produced because already the antibodies are formed in some other body. So our body doesn't form any antibodies. That's why there are no memory cells in this. So there is no long-term protection. This type of immunity is generally called as emergency immunity, when the body is not having time to form antibodies and the virus or the pathogen that has attacked our body is very strong. Example, anti-tetanus injection. We all know that if we are in an encounter with an iron material, rusted iron or anything, if we face an accident or something, immediately we go to the doctor and doctor gives us anti-tetanus injection. What is there in this injection? Antibodies, the already produced antibodies, ready-made antibodies are there. Then there are injections for snake bite. These anti-tetanus and injections for snake bite are artificial passive immunity. Examples of artificial passive immunity. What is the example of natural passive immunity? That is colostrum, yellowish fluid which is produced by the mother in the initial days of lactation. This is the example of natural passive immunity. This mother's milk, the initial mother's milk, colostrum, contains a large amount of IgA antibodies and it protects the infant against various infections. Let's talk about vaccination. As I said earlier, what is vaccination? Vaccination is actually a method in which some antigens, weakened antigens or pathogens are inserted into the host so that the host's immune system can produce antibodies against it. See the definition? It is the introduction of antigenic proteins of pathogens or part or whole of inactivated, you can see here, inactivated or weakened pathogen. Weakened or inactivated pathogen is given to the host body. It is inserted in the host body so as to trigger the host immune system to produce antibodies against that antigen. Now, why vaccines are given? Because vaccines work on the principle of memory. Suppose if weakened organism is entered in our body will fight against it. Obviously, the pathogen is weakened, so our body will produce strong antibodies and it will kill the weakened pathogen. And as the habit of our body, it will preserve some antibodies in the form of memory. And if there is any subsequent attack of the same virus or same pathogen, our body can fight against it. So this vaccination principle works on the principle of memory. The antibodies produced in response to the vaccine will then help the body fight against the actual infection. If there is an actual infection later on in our life, the antibodies produced can fight. So vaccination is very, very important for our body. And generally these vaccines are given before the age of 5 years, so that in later part of life, any time at any age, if there is an actual attack of the pathogen, our body can fight against it. During the actual infection, these antibodies will be produced in large amounts by the activation of memory B and T cells of the immune system, which will recognize the antigen. It will recognize the antigen because it had fought with it in the earlier stage of our life, and then it will form suitable antibodies faster, and the virus or the bacteria or any pathogen can be killed. So vaccination is very important, and it is responsible for providing immunity for the whole life of an organism. Now, along with vaccination, there is another term which is used, which is called as immunization. Immunization is a type of passive immunity. Vaccination is active, whereas immunization is passive. In some cases, if a person has been severely infected or needs antibodies in his body faster to fight the infection, ready-made antibodies may be introduced. The person is not having the time to produce antibodies in its own, and as I said, that production of antibodies will take time. But if the person has been attacked with a very strong pathogen, the person needs antibodies faster. This method is employed when time is insufficient. Here there is no time to activate the host's immune system, because until the host immune system is activated, the virus will be very strong, the pathogen will be very strong, and it will kill the person. New techniques are involved in this, like recombinant DNA technology. They allow the production of antigenic polypeptides in bacteria and yeast. And if antigenic polypeptides are formed here, this will allow for large-scale manufacture of vaccines against specific antigenic polypeptides of various pathogens. This method is still going on. Many scientists are trying to develop this method. As the cellular material of pathogen is not present in the vaccine, side effects of this may be less. Sometimes what happens, because of the cellular content or the genetic material of the pathogen in a vaccine means the weakened organism, it sometimes has side effects. But if recombinant DNA technology is used, it will not have much effect. Now this recombinant DNA technology method will be explained to you in chapter number 11 and 12, when we'll be studying with biotechnology. So no need to go in detail right now. We'll be dealing with this in chapter number 11. Another topic of immunity is allergies. What are allergies? You can see this figure. So many things are given, something somewhere here. This type of mark is there, right? Yes. Allergies means when our body responds extraordinarily. There are some allergens in the environment, like you might have heard that some people have allergy of pollen grains, some have allergy of rose, some have allergy of dust. What happens when these type of materials come in front of that, come in contact with the person's immune system will respond in an extraordinary manner. There will be a lot of response of the immune system. It can also be called as aggravated response of the immune system to certain antigens which are present in the environment. And mind you, these antigens are or these allergens are different for different persons. Suppose if a person is having allergy of dust, the other person along with this person may not have the allergy of dust. So these are different antigens. The substances to which such immune responses produce are called as allergens. And we have discussed earlier that the antibodies produced during allergy are of IgE type. Some examples of allergens are mites in the dust, pollens, animal danders, some fragrances, some type of flowers, and many other things. Even some eatables, like some have allergy of peanuts, some have the allergy of beans. So different types of allergies are there. What happens during allergy? The symptoms of allergy are sneezing. Person starts to sneeze. Sometimes water is released from the eyes. The nose starts, is releasing mucus. And some people may also have difficulty in breathing. Why these allergic reactions occur in our body? Because in our body, when these in some persons who are prone to this allergy, in their body, because of these allergens, some chemicals are released. Chemicals like histamine and serotonin are released from the mast cells. How to prevent the person, how to protect the person from these allergic reactions? Doctors give them some drugs like antihistamines, adrenalines, steroids. They reduce the symptoms of allergy. Now, today's life, the modern day lifestyle has led to lowered immunity. We, or the young generation, they have very weak immunity. And as the pollution rate is increasing a lot nowadays, the people or the children, young children especially, they are having very low immunity, and so they are having increased susceptibility to allergies. They get allergic reactions very fast. Now, another interesting topic of immunity is autoimmunity. As I said earlier, that our body has a capability of differentiating between self and non-self part, right? Like what is our body's part, what are the parts of our body, what are the parts which are not of our body. It has the capability. Now, in some cases, what happens, our body will not be able to differentiate between these things. That means it will not be able to understand whether the part that is present in our body is actually ours or it has come from outside. So what it does, it will start attacking our own body parts. The body will attack its own cells due to some reasons. We actually don't know the reasons till now. There are some genetic reasons or some other unknown reasons are there in which our body will not be able to differentiate between self and non-self. So what it will do, it will try to damage its own cells. It will try to damage its own body. And when it tries to damage its own body, it results into a disease which is called as autoimmune disease. The example of this autoimmune disease is rheumatoid arthritis. You can see in this figure, this is a normal hand. This is the hand of a person having rheumatoid arthritis. The body has tried to attack the bones, and so the bones are deformed. So this is the case of autoimmunity. See, the synovial fluid has degenerated completely and the shape of the bones is distorted. Let's talk about the immune system of our body. The immune system of our body consists of lymphoid organs, tissues, cells, and some antibodies. What are the functions of our immune system? As we have studied till now, it differentiates between self and non-self. It protects the body against non-self or foreign antigens. It acts on the basis of memory and protects the body against antigens to which it has been previously exposed. It is involved in allergic reactions. It is important in autoimmune disease, and it is also important in organ transplant and graft rejection. These 6 points we have already studied. So this is just a summary of the functions of our immune system. Let's talk about lymphoid organs. What are these lymphoid organs and why they are called lymphoid organs? Because they are responsible for production of lymphocytes. Lympho. You can see here. So from here only the word has been used. lymphoid organs. These are actually the organs where origin and maturation or growth proliferation of lymphocyte will occur. Lymphoid organs are responsible for providing protection against foreign antigens. They are also responsible for protection of our body through the spread of tumors. These organs exist in three forms: primary, secondary, and tertiary. Here we are going to deal with primary and secondary lymphoid organs. This is based on the stage of lymphocyte development. It is based on development of lymphocyte. Lymphocytes are in which stage of development, and the organ which is involved here, maturation of that organ. So it depends on that. Lymphoid organs contain connective tissues with different types of leukocytes, or leukocytes means WBC. Out of these leukocytes, lymphocytes are usually present in the highest percentage. It is not important it is in which lymphoid organ. Whatever the lymphoid organ is, lymphocytes, that means one type of WBC, they are present in the highest percentage because they are only the one which are responsible for providing immunity. Let's have a look. See here, you can see all the lymphoid organs, tonsils and adenoids. Here there is the thymus gland, spleen, Peyer's patches, bone marrow, appendix, and lymph nodes. Now, bone marrow and thymus are the primary lymphoid organs. What is their function? Immature lymphocytes differentiate into antigen-sensitive lymphocytes over here. Secondary lymphoid organs are spleen, lymph nodes, tonsils, Peyer's patches of small intestine, and appendix. They provide the sites for interaction of lymphocytes with the antigen, which then proliferate to become the effector cells. So the function of primary lymphoid organs and secondary lymphoid organs are different. Let's see one by one. First, we'll see primary lymphoid organs, that are bone marrow and thymus. The bone marrow is the main lymphoid organ where all the blood cells, including lymphocytes, are produced. We all know that all the blood cells are produced inside the bone marrow. So lymphocytes are also produced in the bone marrow. Second, we'll talk about the thymus gland. It is located beneath the breastbone and near the heart. It is a lobed organ. The thymus gland is quite large at the time of birth. We have studied this in 11th standard, last chapter. Thymus gland is very large when there is a birth of an organism. But slowly and slowly, as the age increases, the size keeps on decreasing. And by the time puberty is attained, it reduces to a very small size. Whereas in old age, it is almost diminished. Both bone marrow and thymus provide microenvironments for the development and maturation of lymphocytes, T lymphocytes and B lymphocytes both. We have already seen this. These lymphocytes are formed in bone marrow and thymus gland. Spleen. It is a very large bean-shaped organ and it mainly contains lymphocytes and phagocytes. It acts as a filter of blood by trapping blood-borne microorganisms. Now, suppose if there are any microorganisms which are present in the blood, and if the blood goes in the spleen, these microorganisms will be filtered out of the blood. It is also a large reservoir of erythrocyte. It is also called as graveyard of RBC. When RBC die, they die and get destructed in the spleen itself. Talking about lymph nodes, what are lymph nodes? They are small solid structures which are located at different points in the entire lymphatic system. And what is the function of these lymph nodes? They trap the microorganisms or other antigens that get inside the lymph and the tissue fluid. Antigens which get trapped in the lymph nodes are actually responsible for activation of lymphocytes. Now here what happens when the microorganisms or antigens are moving in the blood. From blood, they enter into the lymph and tissue fluid. Now, from lymph, they will get trapped in the lymph nodes. When they are present in the lymph nodes, the lymphocytes get some time to check these antigens and to form antibodies and to act against, to get into phagocytosis against these antigens. So, here the lymphocytes will be activated. And that's why when lymphocytes are activated, they will form the immune response. So lymphoid organs or lymph nodes are very, very important for causing the immune response. Lymphoid tissue is also located within the lining of some major tracts like respiratory tract, digestive tract, urinogenital tract, because as I said earlier, that these are in contact with the external environment, and here the chances of attack of the viruses bacteria is more. It is also called as mucosal associated lymphoid tissue, that is MALT. Please remember this full form. It has been asked in board exams many times. It forms approximately 50% of the lymphoid tissue. So 50% of the lymphoid tissue is present in the lining of respiratory, digestive, and urinogenital tracts. So that was all about immunity today. In the third part, we'll be dealing with AIDS and cancer. And then in the fourth part, we'll be dealing with drugs and alcohol abuse. So if you have any doubt till now, you can contact me. And till that, take care, have a nice day, and keep studying.